to chair the session. My name is Manabu Oyama from the Keio University, Tokyo, Japan, and it's my real honor to have a co-chair, Professor Michael Philpott from London. And let's get started. And the first presentation will be given by Professor Philpott, and he's going to talk about the effect of environmental stress on cultured leafy cells and implications for hair growth in vitro. Prof, please.
Thank you. I'd like to thank the organisers for giving me the chance to attend this meeting, and it's been a thoroughly enjoyable meeting so far, and also the chance to talk about some work. Now, the work I'm going to talk about this afternoon is really work that's been carried out by two former PhD students in my department, so they deserve the credit for this work. And we started this project a number of years now, and we were interested in trying to understand what were the signalling mechanisms that may be involved in androgenetic alopecia. And we knew from the genetics that the androgen receptor was strongly implicated, but that didn't really tell us much about the downstream signalling pathways and how that may be resulting in androgenetic alopecia. And so we decided to set up a project where we would look at the double pillar cells from boarding and non-boarding scalp. And I'm going to skip over the background to androgenetic alopecia here, because I think most people probably understand the basics of this miniaturization of the terminal hair follicle to a vellus-like hair follicle. And when we started this project, we discovered quite early on, I was still actually getting down in the lab in those days and doing some experiments, and I'd spent a lot of time dissecting out dermal papilla cells from the intermediate boarding hair follicles that we got from hair transplant surgeons. And I grew up all these papilla cells, and we then froze them down in liquid nitrogen, and we'd done these experiments with matched biopsies. So we had biopsies from the occipital region and from frontal boarding region of the patients. And when we grew these cells up and put them in liquid nitrogen, when we brought them back out of liquid nitrogen, we could always get the occipital dermal papilla cells up. They would always come out of liquid nitrogen and always grow perfectly well. But the boarding ones just would not regrow. And when we were growing these papilla cells, we basically noticed that the boarding ones very quickly in culture actually formed this large sort of almost like fried egg type appearance. And so a PhD student in my lab, Adiam Bata, actually did a nice PhD with me and published a paper in the Journal of Investigative Dermatology in which she showed that these cells were actually undergoing premature senescence, and that this senescence was associated with expression of the P16 incorpore. And so we published in the JOD, and this was interesting because cell senescence associated with P16 tends to be associated with cell stress, oxidative stress, rather than telomere shortening. And so we suggested that the papilla cells in vitro are in some way sensitive to oxidative stress. Now, it is well known that cell culture is a major cause of cell stress. We all grow our cells under 5% CO2, 10% CO2, but 95% air. And 95% air has about 21% oxygen. And that's much higher than the concentration that most cells in the body experience. And so typically dermal papilla cells probably see between 2 to 5% oxygen in their normal environment. And it's known that oxygen or oxidative stress can cause senescence. And so we suggested that the premature senescence of these papilla cells from the boarding scalp was associated with oxidative stress. And so we put forward a theory that the papilla cells from the frontal boarding region, but not from the occipital, were in some way more sensitive to oxidative or environmental stress. And so we decided to investigate this by looking at the effects of oxygen on the cultured cells in vitro. And the way we did this was to get matched biopsies from the hair transplant surgeons. So we get biopsies from the occipital region and the frontal region, and we then microdissect out papilla cells and we grow them in vitro. And when we put these cells in vitro, we grow them under two conditions. We grow them under the standard tissue culture incubator that we all know, and we also use a stem cell chamber. And in the stem cell chamber, we can actually control the oxygen concentration, and we typically grow our cells in this chamber exposed to 2% oxygen. whereas under your standard cell culture conditions, the cells will actually be exposed to about 21% oxygen. And the first experiment we did was just to look at the population doubling time of the boarding and non-boarding propeller cells. Now the axes are slightly different on these two graphs, so I must apologise, but what you see straight away with the boarding propeller cells is that they undergo far fewer population doublings in vitro when grown under atmospheric oxygen compared to the non-boarding. And these are from the same patients. And if you then grow these cells under 2% oxygen, you get a much higher number of population doublings in the boarding, although they never actually, so we get about 12 population doublings here with the boarding compared to 16 in the non-boarding. So generally the boarding propeller cells are much slower to proliferate in vitro, and we've known that from previous publications from Bear Randall's group. What was interesting is when we grow the cells under, very sensitive. When we grow the cells under low oxygen, under 21% oxygen, sorry, and then we switch them to 2% oxygen, you suddenly see the cells take off. So these cells seem to be acutely sensitive to the environment in which you grow them. And we then, with these propeller cells, these same ones that we had in culture, then looked at the effects of oxygen on the migration of these propeller cells. And we found quite strikingly that both at 2% and 21% oxygen, boarding propeller cells always have a much slower rate of cell migration than those from the occipital region, but also they are acutely sensitive to oxygen. So occipital cells under 2% oxygen and boarding, the boarding migrate much slower, and then when you put these cells under the normal cell culture conditions, 21% oxygen, interestingly the occipital propeller cells dramatically decrease their rate of proliferation, but so do the boarding. The boarding cells virtually stop migrating. Okay. We then looked at the rate of cell senescence, and the assay we used here is to actually quantify the senescence-associated beta-galactosidase. So there's a beta-galactosidase expressed at pH 6, which is a classic marker of cell senescence. And this is an assay where we can actually quantify this. And what we find quite strikingly is that our boarding and non-boarding propeller cells under 2% oxygen have a very low rate of cell senescence. When you grow the cells under 21% constantly, the senescence increases and the boarding propeller cells increase significantly more in terms of senescence compared to non-boarding. Strikingly, when you grow your cells under 2% oxygen and then switch to 21%, there's a dramatic increase in the senescence in the boarding propeller cells compared to non-boarding propeller cells. And interestingly, when the cells are grown under 21% oxygen and then switch to 2%, we see very low levels of cell senescence. So the propeller cells from the boarding scalp are much more likely to undergo senescence in culture than the cells from the non-boarding scalp, and they are more acutely sensitive to the oxygen concentration. So we then also looked at reactive oxygen species, and so we quantified reactive oxygen species in boarding and non-boarding propeller cells. And again, under 2% oxygen, there's very little difference in the levels of reactive oxygen species between occipital and boarding propeller cells. But when you put these propeller cells under 21% oxygen, which is the standard cell culture conditions, there's a dramatic and significant increase in the levels of reactive oxygen species in boarding propeller cells compared to occipital, although occipital also increased as well. So we then looked at the ability of cells or some of the enzymes that are involved in mopping up reactive oxygen species. And so we first looked at catalase, and we both looked at the level of expression of protein of catalase and also catalase activity. And again, when we looked at the relative expression levels of catalase, so these are densitometry analysis taken from Western blotting, you can see that even under 2% oxygen, the boarding propeller cells have much higher levels of catalase than non-boarding. And when you put these cells under 21% oxygen, the levels of catalase increase in both cell populations, but again, the boarding cells have a much higher level of catalase protein than in the occipital, and you can see that here in the Western blot with the loading controls here. And when you actually measure the catalase activity, this is more or less effectively reproduced by the level of the protein. So again, with the occipital and boarding, the boarding propeller cells have much higher level of catalase activity than occipital. And when you move these cells to 21% oxygen, again, the level of catalase activity goes up dramatically in the boarding compared to the occipital. Okay? So this would suggest these propeller cells are under an increased level of stress, and that the boarding cells are probably under more stress than the non-boarding. We looked to see whether antioxidant treatment would actually reduce both the levels of reactive oxygen species and also the level of senescence. And so we used NSR cysteine treatment of our boarding papilla cells. And basically here we're looking at reactive oxygen species, and we looked at cells at passage 2 and passage 4. And basically the take-home message is that these are boarding papilla cells, that antioxidant treatment does significantly decrease the level of reactive oxygen species in the boarding dermal papilla cells. And when you measure the level of senescence in these papilla cells, again, it's more pronounced at passage 4 because these cells seem to have built up more oxidative stress or reactive oxygen species. But you can see quite clearly that at passage 4, the antioxidant also decreases significantly the level of cell senescence in these papilla cells. Now that is interesting, and we're not too sure exactly what that means, because classically once a cell undergoes senescence, it's fixed in a senescent state. You can reverse that by changing its gene expression profile. And so what I think is happening here is that these dermal papilla cells maybe are undergoing senescence but are not becoming fully senescent. And that's why at this stage we appear to be able to reverse the senescence in these cells. We then also looked at the effects of both oxidative stress and dihydrotestosterone on secretion of TGF-beta1. TGF-beta1 has been shown to be secreted by boarding papilla cells by Itami and his group in some very nice work. And the first thing we did was just to take boarding papilla cells and non-boarding papilla cells and give them a sub-lethal dose of hydrogen peroxide. And again, you can see quite strikingly that the boarding papilla cells, when exposed to sub-lethal doses of hydrogen peroxide, secrete much higher levels of TGF-beta1 than the non-boarding papilla cells. We then actually then looked at the secretion of TGF-beta1 and beta-2 in response to dihydrotestosterone. And to get a response to DHT, it seems classically you either need to transfect in the androgen receptor into the papilla cells, which is what Itami and his group have done. We've used quite a high dose of DHT here. And what you see here under 2% oxygen is quite interesting. With the occipital dermal papilla cells and also with the boarding papilla cells, DHT actually seems to decrease the level of TGF-beta production under 2% oxygen. But when you grow these cells under 21% oxygen, then dihydrotestosterone actually significantly stimulates the secretion of TGF-beta1. And TGF-beta1 has been shown to be a negative regulator of hair growth. So you get strikingly different profiles in the patterns of growth factor secretion depending on whether you grow the cells under 2% oxygen or 21% oxygen. And there may be a take-home message here for most of us who do all our cell culture work under 21% oxygen. And then again, when we looked at TGF-beta2, the story is a little bit more straightforward. So under 2% oxygen, we see very little effect of dihydrotestosterone on either occipital or boarding papilla cells, but strikingly, in both the occipital and the boarding papilla cells, under 21% oxygen, DHT again stimulates a significant increase in the level of secreted TGF-beta2. We've looked at another couple of growth factors, which I just mentioned very briefly here. The Dickoff, which is a negative regulator of Wnt pathway. This picture didn't transfer across very well. But basically, the orange blocks here are the boarding papilla cells, and the blue are the non-boarding. And so basically, what we see is very little difference in the levels of DKK1 secretion under low oxygen. But when we put the cells under atmospheric, so 21% oxygen, interestingly, the boarding papilla cells secrete much more of this negative regulator of the Wnt signaling pathway than the occipital cells. And also, quite strikingly, IGF-1, which is a possible positive regulator of hair growth, again, is acutely sensitive to oxygen. So under 2% oxygen, you can see here that the occipital non-boarding papilla cells secrete much higher levels of IGF-1 than the boarding, which is probably what we'd expect. Interestingly, when you then put these cells under 21% oxygen, the boarding papilla cells, their secretion of IGF-1 doesn't change, but there's a significant decrease actually in the level of IGF-1 secretion by the non-boarding papilla cells. So again, the concentration of oxygen under which you grow your cells has a dramatic effect on the secretion of growth factors by those cells. And before I finish off, we've started to try to look at the environment of the boarding scalp to see whether it's the hair follicle, whether you see this senescence in vivo, and to see whether it's mimicked in the stuff we see in vitro, whether it's mimicked in vivo. This has been quite hard because the boarding biopsies are quite hard to get. But what we've noticed quite strikingly is when we've looked at, I think, about 5 or 6 boarding biopsies now, we tend to see a lot of P16 in the epidermis and also in the hair follicle of the boarding scalp compared to non-boarding. And we've also looked at BMI1. BMI1 is a negative regulator of P16, and we don't really see too much difference here, although in the non-boarding the P16 is far more punctate in the nucleus of the cells, much clearer, whereas it seems to be more diffuse in the non-boarding. And we've also looked at superoxide dismutase, and again, in the boarding scalp, we seem to see a fairly low level, but a higher level of superoxide dismutase than in the non-boarding scalp. So I am wondering whether the general environment in which the hair follicles in the boarding scalp are living is a more stressed, more oxidatively stressed environment. And so what do we think these studies mean? Well, bear in mind these are in vitro studies, and I think the in vitro model here, we are exposing the cells to an extreme form of environmental stress. And I think what this is telling us is that dermal papilla cells from the boarding scalp, when compared to non-boarding, are far more sensitive to environmental stress, and that this environmental stress is having a marked effect on the function of these cells. Now, whether cells senesce in vivo, we don't really know. But my expectation would be that even if they don't senesce, the data we're showing here is that the boarding papilla cells are far more sensitive to oxidative stress. And I think from the work we did with liquid nitrogen, they're also far more sensitive to any form of environmental stress at all. Now, the boarding papilla cells clearly have a much higher level of cathase activity, but that doesn't seem to prevent the cells from undergoing stress. And so I think in some way these boarding papilla cells are less capable, less able to actually deal with the oxidative stress that they are exposed to. And that's a piece of mechanistic work that we're still trying to understand in the laboratory. And I guess the final question is, we're seeing these really dramatic changes in our cells when we switch them or grow them under 2% oxygen compared to 21%. And as we do all our cell culture in 21% oxygen, should we really be using a more hypoxic cell culture condition? That's one of the questions that we're debating in the lab here, and of course these stem cell chambers are very expensive, so they're not cheap experiments to switch over and do. And on that note, I'd like to say thank you. I would like to thank very much Adrian and Jamie Upton, who did all this work, and also actually a very special thanks to Nilofer and Bessem. The Fargo Clinic basically supports nearly all the hair research in the UK now in terms of providing us with samples and biopsies, and without their support and providing those boarding samples, we would never have been able to get this work as far as we've done it. So great thanks to Nilofer and Bessem. And also thanks to the BBSRC and Unilever who funded Jamie Upton's PhD. Thank you very much.
Thank you very much, Mr. Philpott. I think that we can take just a couple of quick questions. Yes, George?
Hi, Michael. That's very interesting. Is there any evidence that they're functionally different, like any kind of a assay where you're recombining them with a dermal cell?
No, there isn't, George. And I mean, the problem is, in our hands, these boarding papilla cells are very hard to culture, and they senesce very quickly, which is why we went down that route of making some immortalized boarding and non-boarding dermal papilla cell lines, and there was a presentation on that yesterday. And at the moment we're in the characterization stage of those cells, and if those cells do retain their normal phenotype, then perhaps we can do some more work on those cells, because it's very hard to do this work with the boarding cells. It's been very, very difficult.
Any questions? Prof, to what extent do you think this biological difference contribute to the pathogenesis of the AGA? I mean, like, is this a secondary booster of the clinical symptoms or primary events?
I mean, my feeling is it's probably a secondary response. And in fact, I did, after Rod Sinclair's talk yesterday about the pillar rectus muscle and the switch to a adipocyte type phenotype, I did have a quick search last night to to see whether oxidative stress drives a switch in dip sites differentiation. And the literature seems to be a little bit confused on that one. There were a few papers saying that oxidative stress will actually drive a dip site differentiation, and a few other papers suggesting no, it's not, it inhibits. So my feeling is it's probably secondary, but I think it probably plays quite a major role because these cells clearly, in the border modes, clearly are far more sensitive to environmental stress from the work we've done in vitro.
Okay, thank you. Thank you very much. Okay, let's move to the next presentation.